Shri Gurubhyo Namaha. Welcome to our online learning initiative and I welcome you all for the biology class. In last class, we saw about the parallelism between the gene and the chromosome. Now, we are going to step into yet another topic called a linkage and crossing over. So, when I say the term a linkage, we have guessed what it is the close association of the gene. And if the genes are going to be associated closely, how it is going to get inherited. So we'll see that. So our topic linkage and crossing over. Crossing over leads into recombination. That's why the topic called recombination. So first we'll start with linkage. Just now I said a linkage is nothing but a close association of the gene. I'll show you here. It's a close association of the gene or the DNA sequences on the same chromosome. Yeah. Let me draw a chromosome here and we'll see how it is associated. How are we going to denote the gene in the chromosome? So if this is the chromosome, yes, I'll draw the homologous pair and the genes I'm going to represent by a dot. Okay. The dot represents the gene. And already I gave a term. We have discussed about a term called locus. So what is locus? A point, a place where the gene is located is called locus. So I'm going to locate the gene here, okay, which in turn we call it as locus of the gene. And the position 1, 2, 3, 4, we'll number it, 5, 6 and 7. So these are all the position. Locus 1, 2, 3 like that. And the dot represent the gene. Okay. And what is the first point says? It says about the close association. If I say number 1 and 2, you can just look there and check where are 1 and 2. They are closely associated. And if I say 1 and 7, just check out, see the distance, the space. Okay, they are not closely associated, they are quite far from each other. So 1 gene and 7 gene, they have some distance, not closely associated. 1 gene and number 2 gene, they are closely associated. So what happens if they are closely associated? The genes get inherited and they get transmitted to the next generation. That's what we are going to see. So this linkage, I'm going to classify it as two types. One about the closely linked, we call it as complete linkage. Another one where crossing over of the arms of the chromosomes takes place, and that we call it as incomplete linkage. So we'll see. Here this figure represents the location. Okay, as I showed there, the gene 1, gene 2, gene 3, how they are located here. Okay. Now we'll see about complete linkage and incomplete linkage. In complete linkage, I pulled out the gametes and I showed there of a chromosome, a pair of chromosome. So this is one set, another set here, okay, shows, always remember capital letter dominant and small letter recessive character. So both the things are there when the gametes are pulled out and see how it's pulled here, okay. So this, in this, there are no crossing over. The gametes, okay, which is carrying the gene, the same gene going to get inherited and it going to transmit the character. It's a complete linkage. Okay. And next figure, if you see, incomplete linkage, observe carefully, there will be a crossing over of the arms of the chromosome. Already we would have discussed this one in meiosis. In meiosis, stage 1, okay, prophase 1, okay, this will be seen. Okay, let me write here. This occurs in meiosis. Okay. In meiosis, there are two. Meiosis 1 and meiosis 2. First stage it is. In first stage, P1, prophase 1, first stage it takes place. Okay. And in that you can see the crossing over. They look like a bivalent and the arms of the chromosome cross over. Based on that, okay, the genes get inherited, okay, exchange, okay, exchange, and then only it will get inherited. So that you can see here, orange color is one set of the chromosome, Blue color is another set of the chromosome. When it crossed over, again children, keep in mind, crossing over doesn't mean that the arm itself going to exchange. The arms of the chromosome remains same. Okay, let me draw here. Yeah, this is one. Another one. You should have two chromosomes, two sets of chromosomes. Okay, so here these are the chromosome. We discussed this is sister chromatid, this is sister chromatid, and for the red one, the blue one will be the non-sister, non-sister chromatid. We have to say chromosome here because it is in condensed form. So this one, when it intercross, I'm just taking only one arm, that red arm. Okay, let me indicate with the number 1 and this one number 2. Okay, I'm just representing with the number so that you can understand clearly. So the red arm, and then the blue arm, that is another chromosome. That is why you call it as non-sister chromosome. Let me use the same color. Okay, now what happened? It is going to intercross. If you see the blue one of here, okay, and then another of like this. Okay, I'll represent here this one, this arm will intercross, the blue arm will intercross with the red arm, and the red arm will intercross with the blue arm. Okay, the arms of the chromosome get attached and they will exchange the genes there. Okay, that we will represent with the two different colors. Again, make it clear, the arms are not going to get exchanged, only the genes. Okay, and the same thing they have represented here. Can you understand this? Yes, here. Yes. So this you call it as crossing over. It leads into recombination. So now no more it is same. Okay. It is different totally. Okay. In previous case, same it is. Okay. The division also same. Mitotic division, no crossing over. Okay. Just like that the arms will remain same. So the genes get inherited in a same way. But here crossing over takes place. And this crossing over leads into recombination. Because of the recombination, they are all variant now. What is variant? No more similar. There's some variation in some characteristic features. Okay, that leads into all these variations together, leads into a speciation, and that speciation leads into evolution. Okay, formation of new organism. So this is the basic theory for evolution. So, example for this crossing over and then the linkage. Okay, we are going to just imagine if this is the chromosome. In the chromosome, if the gene is located here, already I've discussed there with the diagram. If the gene is located here, another gene is located here. We discussed, right? One, two, three, four. Okay, like the closer they are, very closely associated, and that particular character of the parental chromosome will get inherited to the daughter or son, the next generation. Now, suppose if the chromosome is here, okay, and the distance between the gene, if the distance between the gene is more here, the chances of getting crossing over and exchange of gene will be possible, will be more. In that case, the linkage is incomplete. Okay, so here recombination can take place. Okay, and the same characters cannot be inherited, all because of the chance. By chance, if there is a crossing over and the genes get inherited, I mean the exchanged, okay, then it leads into variation, a recombination one, or else it remains same, but the chances will be too low for this particular character to get inherited to the next generation. And if the genes are too close, like this closely associated, the chances are more, 99.9 percentage we can say, that the characters will get inherited to the next generation. Closely the genes are linked, more the probability for the inheritance. Okay, farther they are located, a less probability for their inheritance. So now we'll see the example, or a familiar example, fruit fly, Drosophila melanogaster. Whenever you think about these genotypes, B, V, G, in the beginning, first class I've discussed, tall means capital T, yellow means capital Y. The same way, black body, okay, and then brown body. Here they are indicating as brown body or gray body. According to our textbook, it is yellow body. Yellow will be dominant, another one black will be the recessive. So this is recessive and this is dominant. They are given the term wild. Wild type is nothing but dominant type. If you see the wings of this Drosophila, normal, and here the wings quite vestigial, functionless. Okay, and these two characters they have taken: one body color, another one size of the wing or the nature of the wing. Okay, these two characters. So it's quite like a dihybrid cross, right? The same dihybrid cross only, but in a different term we are going to see as a linkage and crossing over. How closely they are related, how the parental combination are seen in the F1 generation. Okay, and what are the new characters are seen in the F1 generation. Okay, in Mendel's concept, he intercrossed tall, round, okay, and with a dwarf, wrinkle, and he got a new non-parental combination. In what ratio? That you just guess and keep it. It is 9 is to 3 is to 3 is to 1, right? The same way here, in what way they are getting, why it is so? It is all because of this concept of linkage. If they are closely associated, well inherited. If they are not closely associated, which is not closely associated, the genes, then it is not inherited. And non-parental characters happen to be seen here. So here you can check black, vestigial and wild type. Okay, and we are representing the chromosome separately, like if it is a two character, four gametes will be there, one character, two gametes will be there. Yes, likewise here two characters, so four gametes they pulled out, for different sets of gametes you will get. And here if you take it, it is totally recessive, homozygous recessive. And here however you pull four gametes, all the four will be the same, small b small vg. When you intercross this, you will get the combination like this. Okay, the parental phenotype. You can see the ratio or the frequency is more, but here it is quite less. It says that the genes are closely associated. That is why we have more number of the same parental characteristics. Yes. We'll move to the next topic, recombination. Okay, a new combination is going to emerge. So what is that recombination? It is a process by which pieces of DNA are broken and recombined to produce new combination of alleles. Okay, they are going into the molecular level. So far we were discussing the two chromosome intercross and the genes going to get exchanged. If you go in depth, what is there in the chromosome? The DNA. In DNA, what is there? The gene. That's it. So here they are talking about the DNA. Let me draw the structure of DNA. It's a double helical structure. Yeah, and what happened here? In by which a piece of DNA are broken and recombined to produce new combination. So here if you take a piece of DNA will be broken, another set, okay, of another gene is coming and intercrossing, right? That particular, if this is the condition, there is another, that exchange gene. Okay, and this will get incorporated here, and if you see the structure, it will look like this. Yes. So this technology, it is basically happening in our body. Okay, this technology they are doing in rDNA in biotech field. We call it as rDNA technology, recombinant DNA technology. We are going to see this later in biotech lesson. Okay. So this recombination process creates genetic diversity at the level of genes, okay? And that gene slowly to the DNA, then chromosome, then the cell, then the whole body. So there is a variation. No one is alike, no one is similar. You leave identical twins. I'm just talking in general, right? There are variation that reflects different in the DNA sequences of different organisms. Yes. So you can see the process, meiosis process, how the crossing over takes place. Okay. They are indicating by the color. It says that there is a breakage in the DNA and there is some collaboration in the two different DNAs. Okay. And that leads into like this. Yes. Now, we need a device, okay, for measuring. I'm saying that the gene is located here, okay, another gene is located here, okay. I cannot say for all the chromosome, for chromosome 1, chromosome 2, the gene is located in that place, this place. We cannot say like that, okay. We have to say exactly where it is. We have to measure how far it is or how close it is. So how can it be done? It can be done by this method, gene mapping. Okay. To honor Mr. Morgan, they are using the unit as Morgan unit. The distance between the gene, they are saying it as centimorgan or millimorgan or Morgan in simple. So, gene mapping describes the method used to identify a locus of a gene position where it is. Okay, the position we can say it is so far like 5 centimorgan, 6 centimorgan, like that we can say with the distance. Yeah, with distance. The essence of all genome mapping is to place a collection of molecular markers onto their respective position on the genome. This part, okay, we are going to discuss in molecular biology, okay, because about the codons, selectable markers, all we are going to discuss there. Okay, let us see only about mapping here, the gene mapping. So I'll show you the picture. Just check in the gene map if this is the chromosome, okay, cytogenetic map we call it as, and this is the physical map for our reference. We are saying like 1 liter milk, 2 liter milk, like that. This is 25 morgan or 60 centimorgan, like that. It's a physical unit we call it as, okay. And this MB represent molecular bases. Molecular base is nothing but the base pair. Already I've discussed about the 4 codons. That is nothing but adenine, guanine, cytosine, and thymine. You can see here the DNA sequences A, T, C, G. So in this particular space, what are the sequences? They'll pull out and check. Okay. So this is a mapping method to check the position, I mean the locus and how far they are located. Okay, in a glimpse on the whole, genetic map is nothing but it is show relative order, distance from each other based on their pedigree studies. Pedigree studies, the family chart. Okay, the chances of now you can see here the letters A to H. Okay, and highlighted the colors also A, C, E, F also. A and C first, if you take it, is higher than the chance of chromosome breaking between A and B. So who is closer? A and B is closer or A and C are closer? Just have a look. So this is closer. The chance of breaking A and B is quite less compared to the chance of breaking A and C. Okay, because they are far away. A and C are far away. And E and F, if you see, it is quite far. And the same, if you see F and G, okay, they are closer. Same thing. Breaking between F and G is too low. I mean, the chance is too low, and the breakage between G and E is quite more. Okay. So, on the whole, we discussed about the linkage, crossing over, and genetic map. Next class, we'll see about mutation. Thank you.